Thank you so much to LabRoots for inviting me to give a keynote presentation today, and to Marilyn Rusnick for thinking of me and suggesting me to LabRoots. Today, I'd like to talk about the work of my laboratory and many other laboratories investigating the role of viruses in natural ecosystems. So, billions of times a day, this process of a virus finding its microbial cell, in this case we have a zoomed in view of T4 finding its specific receptors on the surface of E. coli, and then this fantastic molecular machine delivers its genome into E. coli, piercing through the bacterial cell wall, which is a non-trivial task, as any of us who work on bacteria know. And the DNA of T4 quickly replicates, and within 20 minutes, 200 viral particles are produced. And so this very process is happening throughout natural ecosystems everywhere. And our questions are, how is that impacting ecosystems? And one of the things that really started the field was the discovery that viruses are extraordinarily abundant in natural ecosystems. Typical seawater sample, which is shown in the upper left-hand corner of the slide, this is a transmission electron micrograph, shows all these virus particles as well as a bunch of bacterial cells. Typically, viruses outnumber cells by a factor of 10. The image at the lower left, produced by a colleague, Jed Furman, at the University of Southern California, shows an epifluorescence microscope image. You can see in the middle a pinnate diatom, some bacteria, but that cloud of small particles in the white circle are virus particles. A really fun website called What If took all the viruses out of the ocean, would form a pile of viruses that would dwarf New York City. There's been estimates that viruses number 10 to the 31 on planet Earth, which is far more than the age of the universe or even the number of observable stars in the universe. And, you know, interestingly enough, our own genome is 8% viral. Another interesting thing is that viruses are really unknown. Their genetic composition is extraordinarily unknown. We have a bioinformatics pipeline known as Virome, and in Virome we have attempted to take all of these predicted open reading frames, predicted genes, and classify them according to whether they have a known function or an unknown function. And what you can see is that in the vast majority of the sequences that we see in viral metagenomes have homology to only other unknown sequences in the environment. This is extraordinarily high for soils. 90% of open reading frames are only seen in the environment, never been seen in a known organism. So we have a lot of unknown, we have a lot of viruses, but surely among the top 100 most abundant genes, we must know something, right? And so this diagram shows a clustering of predicted peptides from viral metagenomes, and it's rank ordered. These are the top 100 most abundant clusters from a number of different viromes. And we thought, well, surely we know most of these, right? Well, not really. So it turns out that that proportion, that high proportion of over 60% of the genes being unknown, is true for even the top 100 most abundant peptides, predicted genes within viral metagenomes. And this problem is pretty astounding when you think about the amount of sequence data we have now available largely through metagenomics from viruses. So this plot shows the extraordinary growth in the number of uncultivated viral genomes. As of 2018, it was around 700,000. I think it's probably well into the millions now, given the amount of sequencing that we've done. So really, as with a lot of modern biology, getting genome data is easy now. The problem is getting understanding from all of this sequence data. So to start off, I'd like to just give three sort of examples, some vignettes about what these unknown and extraordinarily abundant viruses are doing to ecosystems. And so, three ways in which marine viruses affect you: fishing, rain, and oxygen. So what are you talking about? So first of all, viruses and good fishing. So the western edges of continents tend to have great fishing, and it's largely because of physical oceanography. These are upwelling zones. And so here you can see on the left-hand panel, the upwelling zone off the eastern United States, and the upwelling zone off the western coast of South America. These upwelling zones are extraordinarily productive, supporting very large fisheries, and also these upwelling zones have outsized impacts on the global carbon cycle. So it turns out that the most limiting nutrient for phytoplankton in the ocean is iron. The difference between the concentration of iron inside a phytoplankton cell and outside a phytoplankton cell is about 19,000 to 1 in the ocean. And so a colleague of mine at the University of Tennessee, Steve Wilhelm, asked the question, well, what if that source of iron matters? And so what this plot shows is the best source of iron, meaning the most bioavailable source of iron, is from a viral lysate, and not some of the other forms of iron that we use in growing phytoplankton. In culture, like chelated iron with EDTA. And so that was a very interesting observation. And so Steve's lab went and did some calculation of the mass balance of iron necessary to support the productivity of the Peruvian upwelling zone. And they concluded that up to 80% of the demand for iron for phytoplankton growth in the Peruvian upwelling zone was actually supplied through viral lysis. So this idea that viruses make available trace nutrients for more growth is an important ecosystem consequence of viral lysis. So viruses make rain. This sounds a little extraordinary, but in high latitudes around in the northern hemisphere, around the United Kingdom, and a belt going across, I guess about sort of 50 to 60 degrees north, maybe as deep as 40 degrees north. And there's another band around the southern hemisphere. This organism, or this group of organisms called coccolithophores, can bloom in tremendous numbers. And so what you're seeing in this image is the southern coast of England and this milky seas phenomena, where these coccolithophores, which have these calcium carbonate shells on the outside of the cell, which you can see in the upper left-hand part of the figure, form. And so when these organisms bloom, you literally get an enormous amount of calcium carbonate. And this carbonate actually sinks out and has contributed to massive geological objects like the White Cliffs of Dover. Well, it turns out that this coccolithophore has a virus, actually a giant virus. And a colleague of mine, Willie Wilson, in the UK, asked a question of, when the viruses lyse the cells, what happens? And so one of the things is coccolithophores and many phytoplankton have to have osmoprotectants. And one of these osmoprotectants is dimethyl sulfoxide propionate. Well, when the viruses lyse the cells, that osmoprotectant is released. It's processed by bacteria into a volatile sulfur compound known as dimethyl sulfide. Dimethyl sulfide is what really gives the sea that sulfury kind of smell. And the sulfate aerosols are very important in forming cloud condensation nuclei, which is actually a negative feedback on the growth of more phytoplankton because it forms more clouds. And Willie actually established this. What this plot shows is the growth of an E. huxleyi culture, which is the pink line, and then added viruses, which is the blue line. And then the red line is a pulse of dimethyl sulfoxide that comes out of the culture once viruses peak. And that's when you're getting peak levels of lysis of those cells. And this, so how important is this process? Well, it's second to the most important process, which is actually grazing of the coccolithophores by things like copepods. And it's actually even more important than just the lysis of autolysis of the cells themselves. The last little vignette here is, if you're breathing, thank a cyanophage. And we're all breathing. So cyanophages are phages that infect cyanobacteria. And what you see here on the left is a dividing Prochlorococcus cell. And these cyanobacteria are abundant in regions of the ocean where really there's not a whole lot going on. And so what I have circled are the oceanic gyres in the image on the right, and that's the domain of Prochlorococcus. And Prochlorococcus, like many microorganisms, have viruses that infect it. And it was discovered by Nick Mann and also Debbie Lindell that actually, curiously enough, these cyanophages infecting Prochlorococcus carry photosynthetic genes. And the reason they do this, we believe, is that the photosystem turns over very rapidly in the cyanobacterial cell. And anything that interrupts protein synthesis, like growing a new virus, will interrupt the production of these rapidly turning over photosynthetic proteins. And so literally the virus is, the idea is that the virus is sustaining the photosystem during infection so that it can make more cyanophages. And given the levels of infection occurring, and the fact that these cyanobacteria provide about 50% of the oxygen in the atmosphere, if we, rough numbers say that about 10% of these cells are likely infected by a cyanophage containing photosynthetic genes, then it's possible that as much as 5% of global oxygen production actually comes from cyanophages and is sustained by cyanophages. So that's 1 out of every 20 breaths. So hopefully I've convinced you that these viruses of microbes have pretty substantial ecosystem impacts. We've been reminded through the COVID pandemic that viruses change host behavior, which indeed changes ecosystems. And in this case, it's humanity. And so the lockdowns that occurred in China and India and are currently occurring just about everywhere have had a tremendous impact on air quality. These graphics show on the left the level of nitrogen dioxide, a potent air pollutant in China before and after the lockdown. And then just the pictures in New Delhi of the air quality 1 year later after the pandemic is pretty striking. So what are viruses doing? Why do they matter? And what's the big picture questions that my lab and several others pursue? First of all, they move nutrients around, they influence productivity, and that, like for instance, the fishing story, because they're releasing micronutrients, but really by how much is this process important? We know that in the Peruvian upwelling it is extremely important, but what about other ecosystems? We also know, I didn't talk about this a lot, but viruses move genes around, especially important for clonal organisms. And so which genes are most moved around, and why are those genes moved around through horizontal gene transfer and using viruses as a vector? They also, virus host interactions are very specific. And so how would, on a community basis, that specific process of viral infection influence the composition of the host community? And what are those specific effects? We also know that when a cell is infected, its biology is very different. So what are those specific effects on hosts? And how do all of these emerge to have impact on ecosystems? So these are really the second, third order questions that are driving us. The approach that we use or metagenomics has become extraordinarily important to approaching these questions. And the driving hypothesis of my lab is that genome to phenome connections will really drive us towards answering these questions. So what I've got in the graphic here is that we specifically isolate viral DNA from ecosystems and then sequence that in metagenomes. And the question I want to know is to identify viral populations within that community and then understand something of their biology, understand something of their life history, as we say as ecologists. And those life history features are really to do with the viral infection process and viral host interactions. So what is the burst size of the virus, its latent period? What kind of life cycle does it have? Is it lysogenic? Is it lytic? Is it pseudo-lysogenic? Is it chronic? What is the host range of the virus? How many different host populations could it infect? How fast is the virus lost from the system? This is a very important consideration. And lastly, how infectious is it? And I think as COVID has taught us, these are big questions for viruses. And the more we understand, the more we can model any sort of epidemic. And really, all of those summarized come together to understand how viruses change host biology and ecology, and this will help us to understand those emergent impacts that I've talked about. So an approach that we've taken is to really focus on the few genes that we can identify that are abundant and that we know something about. And so we focused on peptide and nucleotide synthesis genes. I'm only going to talk about the nucleotide synthesis genes today, but we did discover, and this is the graphic I showed earlier, that DNA polymerase is a very abundant enzyme, ribonucleotide reductase, and GRO-ES is a chaperonin, or it's the small subunit of the chaperonin. We have actually found the large subunit chaperonin. I won't talk about that today, but all of these are found in viruses. They're critical to phage biology. Nucleotide synthesis is at the core of what a virus needs to do. And this is especially important for lytic viruses because they need to quickly infect and then produce and find the next host. There's also, intriguingly enough, many of these genes are obviously shared between cellular life and viruses. So we can start to see those deep phylogenetic connections and the differences in selective pressures on these genes in viruses versus cells. And really, those phylogenetic analyses, I believe, are the foundation of the first step towards greater biological understanding. And so we've looked at known phages and asked, well, just how abundant are these genes? Because the thing about viruses is they're polyphyletic in their origin. So there's no single marker that's going to capture all of viral diversity, no single gene. But if we can find some common genes, they may tell us something interesting about evolution and biology. And so it turns out that family A DNA polymerases are very common among known viruses. 26% of known viruses have them. Ribonucleotide reductase is also very common. It may actually be more common in the unknown viruses in the environment than actually in our cultivated representatives. So the first work I want to talk about is our work with DNA polymerase A, and these three students contributed an enormous amount of work to this project, our ongoing project. So Pol A is, it occurs in bacteria. In bacteria, it's a relatively low processivity polymerase. It functions in repair, it removes primers, fills in Okazaki fragments. It's not the workhorse for cells. However, in phages, it is their primary replicase. And it has a very different biochemical characteristics in phages. So for instance, in phage T7, which is the one we know the most about, has a very high catalytic efficiency. Intriguingly enough, we wouldn't have sequencing today without Pol A because it was the basis of the first sequencing enzyme. Helen Schmidt was an undergraduate in my lab and happened to be also a biochemistry major, which helped a lot with this project. And Helen discovered, first of all, looking through our metagenome samples, typically what the graphic at the top shows is that in viruses, this is a smaller enzyme. The 5' to 3' exo is not present. That's the proofreading part. And the known viruses tend to have smaller versions of the gene. This is not unusual for viruses. They tend to, whatever gene they have, they tend to make it small and efficient. And Helen noted that there was a motif where a particular, this is a motif intricate to the active action of the polymerase, that a single mutation in motif B is responsible for pretty dramatic changes in the enzyme. Now, this is known from in vitro studies with the T7 DNA polymerase. So if the wild type in bacteria, all bacteria have the phenylalanine 762 position. If that phenylalanine is mutated to a tyrosine, there's a dramatic increase in the catalytic efficiency. And this even is the mutation that allows for the incorporation of dideoxynucleotides for sequencing. If you change that to a leucine, the efficiency goes down, but the fidelity goes up. So this is very, very interesting. And Helen had, this slide shows a phylogram of our viral polymerases as well as bacterial polymerases. And so the bacterial polymerases from a vast diversity of bacteria, scanning from Synechococcus or from Prochlorococcus all the way to Thermus aquaticus and E. coli, fairly monophyletic. But as we look at this same enzyme in viruses, we get this explosion of diversity. And I'm pointing out three specific subclades here. The blue subclade is our T7-like clade. It turns out all the polymerases in this clade contain the 762 mutation. Then the red one, which is sort of like our type strain there, is colophage T5, had the phenylalanine mutation. We're calling that neutral. And then the purple was comprised almost entirely of environmental sequences, and that contained the 762 mutation. There was a distantly related phage of an alpha proteobacteria that was actually a pseudo-lysogenic phage. And so looking at this, and I won't show this data, but Helen went and looked at all known phages, and it turns out that known phages having this enzyme, it looks like these different clades are actually linked with phage life cycle. So the upper two, the blue and the red, appear to be only in lytic phages, whereas the purple one appeared to be largely in lysogenic and pseudo-lysogenic phages. And so here we may have, our hypothesis is that a single mutation in pol A may be linked with the life cycle of unknown viruses. And what I'm talking about here is the lytic or lysogenic life cycle. So shown on the left is the lysogenic or pseudo-lysogenic life cycle, where the phage actually enters this phase of being carried by the host and may or may not produce viruses eventually. And then the lytic, where you get an infection and then quick replication and lysis of the cell. One thing that has been an extraordinary help to us in all of this work with replication proteins and with chaperonins has been long reads. And so we have a library that a viral metagenome library that we sequenced using PacBio and Illumina. And as this table shows, what I want to point out here is that we get a far higher yield of full-length polymerases from hybrid assembly of PacBio reads as well as Illumina reads. And this is very important because it is a very large protein, upwards of 700 amino acids in size. And so as the more we can apply long-read technology to metagenomes, the more we can really build these genome to phenome hypotheses. Later work done with one of these long-read libraries, the Circ library, found an extraordinary amount of pol A diversity within the Chesapeake Bay. And so what this slide shows, this Venn diagram shows, is the Circ library, which is in blue, the blue circle over on the left, and a more traditionally Illumina, or actually this was 454 sequenced metagenome libraries, viral metagenome libraries, known as Metagenomes Online, a database we maintain. And as you can see, the 2/3 of known pol A viral diversity was actually found in one Chesapeake Bay sample. So I think if you want to get a deep sampling of viruses on Earth, go to the Chesapeake Bay. It is a highly diverse microbial ecosystem. And as you can see, the small balls within the Venn diagram are actually clusters of pol A peptides. And you notice that the colors are pretty exclusive. And so it turns out that these markers, this single mutation actually links with large 75% clusters. We get very few mixed clusters, and which indicates to me that this particular mutation is extraordinarily important in driving the evolution of the entire molecule. We've also gone out and looked at the gene neighbors of pol A in hopes of gathering more information about the replication module itself. And as we've loaded on more sequences, we've actually seen some of the clades actually split. The phenylalanine group has split into two. The leucine group is split into two. But that tyrosine group, and that's all in the tree shown on the left, has maintained its clading and its sort of monophyletic nature. And so one of the interesting things that we started to look at, well, what is around pol A? And could that help us understand its biochemistry and its impact on the biology of the virus a little bit more? And so when we look at the tyrosine clade, it was very interesting, both the tyrosine and the phenylalanine clade, is that they also contained ring-shaped helicases, which are the helicase unwinds the DNA, and they were highly processive, fast helicases that were associated with those phenylalanine and tyrosine pol A's, which is what you would need for a very fast replicating polymerase. Another thing that was interesting is we also found ribonucleotide reductase associated with those same pol A's, as shown in the lower half of the diagram. And we almost never saw R&R with the leucine pol A's. So why is this interesting? Well, it turns out that RNR is the only enzyme that can actually process ribonucleotides into deoxyribonucleotides. And of course, deoxyribonucleotides are the essential substrate for pol A. So if a virus carries an RNR, it's actually controlling the availability of the substrate for DNA synthesis, which would be, again, very important to a lytic virus, which needs to produce lots of DNA and do it very quickly. In contrast, when we look at the top of the leucine pol-As, those are the ones that are slower but have higher fidelity. They don't have RNR. And so it's possible that these are lysogenic viruses that it's much more nuanced. They are actually controlling the host, utilizing the host machinery for making deoxyribonucleotides. They replicate slower, but their fidelity is better. And so there's these trade-offs that we think we're seeing. So I'm going to transition here and talk about RNR a little more in depth. It is an ancient enzyme, likely responsible or at the transition between RNA world and DNA world. It comes in a number of different biochemical classes I'll talk about in a minute. It regulates the available pool of deoxynucleotides for DNA synthesis. It increases the rate of DNA synthesis tenfold when it's present in a viral genome. There's some thinking that it may have a role in phosphorus recycling in nutrient-limited environments. Of course, phosphorus is very important in DNA synthesis or nucleotide synthesis, period. And we find that it is very common in soil and in viral metagenome libraries, probably more common than even we see in known phages. One of the interesting things about ribonucleotide reductase, of course, anything having a DNA genome needs this enzyme. So that would include all cellular life. So all cells have this enzyme. It has a pretty complex biochemistry that links with environmental conditions. So that's always interesting to an environmental microbiologist like myself. It comes in three classes. There's class 1, which is the oxygen-dependent version of the enzyme. Class 2, which is oxygen independent, but is dependent on B12 cobalamin as its cofactor. And then an oxygen sensitive version, which is found only in anoxic environments. And really, class 1 has the most complexity. It also has different metallic cofactor requirements, which is another interesting thing, knowing the importance, as we do, of micronutrients, particularly iron. So if you'll notice here in that metallic cofactor column or row at the bottom, it can use manganese, it can use iron, or possibly none. And so when we look through the environment, do we see links between the types of ribonucleotides, RNRs that are carried by viruses and the prevailing environmental conditions? There has been, as I mentioned earlier, an explosion in the availability of viral metagenome sequence data. And one project that has contributed an enormous amount of data has been the Terra Ocean study. And Amelia Harrison, my student, has looked deeply into these libraries and uncovered, this is an old slide, we're now up to 20,000 something sequences out of RNRs out of viruses. And we've been able to demonstrate entirely new possibly new biochemical classes of RNRs. And what this is pointing out that in the Terra Oceans data, which is largely the surface ocean, maybe down to a few hundred meters, definitely an oxygen-rich environment, only 4 class 3 RNRs were observed out of literally tens of thousands of sequences. And so certainly the availability of oxygen is certainly shaping the RNRs that viruses are carrying. And Amelia discovered something something I think is very interesting, that among the class 1 RNRs, we think we see a dramatic split between the class the class 1 RNRs, I apologize, the class 1 RNRs that are seen in cyanomyoviruses, which look like T4, the one up at the top there that has the long tail and the long tail fibers, versus cyanopodoviruses, which is the one shown more in the center of the slide with the short little tail. And these two groups of viruses tend to have very different genome sizes. Cyanopodoviruses have smaller genomes. Cyanomyoviruses tend to have large genomes, upwards of 150 to 200 kb. And we see them in distinct groups. Another interesting thing was that the cyano-SP class 1 enzymes lacked regulatory domains. And these regulatory domains are the domains that actually negatively regulate the activity of RNR so that not too many nucleotides are produced, that it matches the level of DNA replication going on. Well, the class 1 enzymes in cyano-SP phages lack those domains, which again would make sense for a lytic virus that's just bent on producing more DNA, producing more viruses. We also do see class 2 enzymes in class 2 RNRs in cyanophages as well. And interestingly enough, cyano-M and the cyano-M clade, the cyano-SP clade, those are class 1 enzymes, whereas the host, the Synechococcus host, actually carries class 2 enzymes. So their viruses are even going in a different direction with the biochemical classes as opposed to their hosts. And why is it interesting, these two different viruses, cyanomyoviruses versus cyanopodoviruses. And this slide shows a time series of four viromes that we collected in the Chesapeake Bay over a 24-hour period. And Eric Sikowski, a former student in the lab, looked at the prevalence of these different groups according to their RNRs over time within the libraries. And it turns out that there really is an inverse relationship between the abundance of these genes over time. So it seems like the myoviruses are, quiet and infecting cells while at the beginning, whereas the podoviruses are producing cells, are producing viruses, and then they swap places. So this may be an example of niche exclusion because both of these viruses are infecting similar hosts. So our next question was to actually link RNRs and POL-As. And so what this slide shows is a concatenated tree of RNRs and POL-As. And what you can appreciate from this is many hundreds of contigs that are plotted here, is that when we have a branch, which is the branches are colored according to the RNR, they tend to be associated with just one type of POL-A, either the phenylalanine, tyrosine, or leucine. Most of the clades behave in this way. And so is it possible that these clades, like clade 4, clade 3, clade 6, share common life history strategies? And so those are the questions that we're continuing to pursue. And so really, I think to summarize, what we're trying to do is really is to bridge that gap. Evolution, when we look at a tree, we're looking at an evolutionary hypothesis, but what we're trying to do is link that to the ecology of the virus and the viral host system and the biology of the viral host system. And so what we have in abundance right now is the genome data. What we're really lacking is the phenome data to tie together with those genome hypotheses of viral life cycle. So along with colleagues at the University of Delaware, University of Rhode Island, the University of Nebraska, and the University of Hawaii, we started into a large NSF-supported study to test genome to phenome hypotheses in viruses of microbes using high throughput phenotyping approaches that we're developing using microfluidics. So really the bottleneck, I believe, in many of these questions is the amount of phenotype data that we can gather. And so we're hoping that microfluidics can provide something of a means of improving that throughput. And, the idea is something like this, where this is what I'm showing here. On the left is a dichotomous key that Eric and Dan developed, where literally you start with the pol A group, and then you start to look outward from there at the combinations of things that you see, and then link those to individual groups on the clades on the tree. And so what this dichotomous key is showing is that if we have a tyrosine 762 or a phenylalanine 762 with certain other gene connections, we would predict that these are putative lytic potoviruses. Whereas if we have a leucine 762, we predict maybe putative temperate potoviruses using these connections. And sort of the grand vision here is to get to something of maybe a rule book for connecting genome to phenome. And here I show on the left genes that are predictive of the biology and ecology of viruses, like nucleotide metabolism genes, possibly structural genes, or auxiliary metabolic genes such as the photosystem genes I mentioned earlier. We've also seen metabolic genes associated with the sulfur cycle or phosphate availability. And then to link these to the biological and ecological features of unknown viruses, which is shown on the right. These are the types of information that we want to really fill in the gaps and to have in much greater abundance. And that's a goal of our current NSF project. So in closing, I would like to acknowledge many folks over the years who have contributed to this work. We've had the fortune of going to lots of different environments and looking at soils and sediments. Much of our work has been in oceanic and marine systems, but we've also had the fortune of working at deep sea hydrothermal vents. Had a few projects looking at the human microbiome and the cattle microbiome. We also have a pretty vigorous bioinformatics effort in the lab, and that's the team Virome that I mentioned over in the upper right. And we've also fortunately been sponsored by many generous sponsors, including the HHMI, the National Science Foundation, the Gordon Betty Moore Foundation, the USDA, and the Delaware EPSCoR program. So with that, I thank you all for your interest and look forward to your questions.